Bismillahirrahmanirrahim. The topic of today's lecture is nucleotide and their chemistry and structure. Nucleotides basically they are mainly composed of purines and pyrimidines. Purines and pyrimidines, if we define them, they are heterocyclic compounds. What are heterocyclic compounds? The compounds which contain other atoms in addition to carbon. Purines and pyrimidines are further classified into two categories. Purines consist of adenine and guanine, while pyrimidine consists of cytosine, thymine, and uracil. Now, here is the basic structure of purine and pyrimidine. There is difference between these two nitrogenous bases. Purine consists of two-membered ring, while pyrimidine consists of single-membered six-carbon ring. In purine, the numbering of atoms start from this position. This is 1, 2, 3, 4, 5, 6, 7, 8 and 9. While in pyrimidine, the numbering of atoms start from this position. This is 1, 2, 3, 4, 5 and 6. Now you can see there are four nitrogen atoms in purine at position 1, then position 3, then position 7, and then position 9. And in pyrimidine, there are two nitrogen atoms at position 1 and at position 3. These purines and pyrimidine, they act as weak bases. It means they can donate their hydrogen atom from the amino group, and they both are present in DNA and RNA. Now, what is DNA and RNA? Deoxyribonucleic acid and ribonucleic acid. They both are the components of nucleic acid, and you know they are the hereditary material of human cell. And these purines and pyrimidines, they are the nitrogenous bases of these DNA and RNA. If they are present in DNA, they are of four types, and if they are present in RNA, they are also of four nitrogenous bases which are present in RNA. Now there is a structural difference in these purines and pyrimidines. Uh, as I already told that they are present in DNA and RNA, and purines adenine and guanine. अभी आप यहाँ पे थोड़ा सा इसका स्ट्रक्चरल डिफरेंस देख लें MCQs पॉइंट ऑफ व्यू से. एडिनिन नंबरिंग के हिसाब से जो मैंने पीछे आपको नंबरिंग बताई है उसके हिसाब से देखें यहाँ पे पोजीशन 6 पे जो है एक अमाइनो ग्रुप है. And this is the structure of guanine. In guanine, at position 2 and at position 6, there is an amino group and there is a carbonyl group. This is thiamine, cytosine and uracil. In thiamine, there are two carbonyl groups at position 2 and 4 and then 5, and then there is cytosine. Cytosine, they have a carbonyl group and an amino group at position 2 and at position 4, while in uracil there are two carbonyl groups at the second position and at fourth position. What is the difference in DNA? There is adenine, guanine, cytosine and thiamine. nitrogenous bases, while in RNA, uh, in place of thiamine, it is uracil. Rest remains the same, that is adenine, guanine, cytosine and uracil. आप इसको लेटर नंबर से रिप्रेजेंट करते हैं इन बुक्स. इट इज़ रिटन इन एडेनिन represented with A, guanine with G, thiamine with T, cytosine with C and uracil with U. Now there are some definitions regarding nucleotide chemistry. Nucleosides. What are nucleosides? These are N-glycosides. Nucleosides, if we define them, we will say that they are derivatives of purines and pyrimidines that have a sugar linked to the ring of purines and pyrimidine. अगर आप इसको थोड़ा easy और आसान ज़बान में डिफाइन करें तो आप कहेंगे कि न्यूक्लियोसाइड्स दे आर composed of purine, pyrimidine plus sugar. What kind of sugar? Ribose sugar. You have studied carbohydrate chemistry and you have studied ribose sugar. It is a pentose sugar, 5 carbon sugar, and it is attached with the purines or pyrimidine nitrogenous bases through the nitrogen atom. तो हम कहेंगे कि न्यूक्लियोसाइड कंसिस्ट ऑफ शुगर प्लस नाइट्रोजनस बेस. नाइट्रोजनस बेस और शुगर अगर एक दूसरे के साथ एडेड हैं तो आप इसे न्यूक्लियोसाइड का नाम दे देंगे. दिस शुगर एंड nitrogenous base is attached with each other via nitrogen atom of that purine or pyrimidine. If it is present in DNA, then this ribose sugar will be 2-deoxyribose, and if it is present in RNA, then it is D-ribose. So similarly, they are called as deoxyribonucleoside and ribonucleoside. Now, these sugars are linked, as I already mentioned, with the purines and pyrimidine via nitrogen atom. The bond which is present, this is glycosidic bond. You have studied glycosidic bond in carbohydrate chemistry. This is elaborate structure from which you can understand that I have already mentioned that this is ribose sugar and this is a purine, adenosine, and it is attached with the ribose sugar via N9, the nitrogen atom at 9th position, and this is guanosine structure in which again ribose sugar is attached with nitrogenous base of guanosine via N9. And when you talk about pyrimidine, they are mainly attached with nitrogen atom at position 1. That is the ribose sugar which is attached with the pyrimidine nitrogenous base at N1. तो आप कह सकते हैं कि ये purines, they are usually attached with ribose sugar via nitrogen at position 9, and pyrimidine, they are usually attached with the ribose sugar via nitrogen at position 1. And now again there is a definition, main definition of our topic, that is nucleotides. If you define nucleotides, then nucleotides, they are nucleosides with a phosphoryl group esterified to a hydroxyl group of the sugar. यानी कि आपका जो न्यूक्लियोसाइड है जो आपने पीछे स्टडी किया है उसके साथ अगर एक फॉस्फेट ग्रुप की एडिशन हो जाए वाया एस्टेरिफिकेशन तो आप कहेंगे कि वो न्यूक्लियोटाइड है. अगर आप फिगर को देखें देन यू कैन सी क्लियरली दैट दिस इज नाइट्रोजनस बेस, it is attached with the ribose sugar. This is called as nucleoside. And when this structure is attached to the hydroxyl group of the sugar with the phosphate group, this is called as nucleotide. If there is one phosphate group, you will call it as monophosphate. If there is there are two phosphate group, you will call it as diphosphate, and if there is third phosphate group, it will be called as triphosphate. All the nucleotide purines and pyrimidines, that is adenosine, guanosine, cytosine, uracil, thiamine, they are present in their mono, di and tri form in the living system. And again, nucleotide consists of nitrogenous base, a ribose sugar, and a phosphate group. Now again, the nitrogenous base is attached uh, via position 1, N1 or N9 if it is purine and pyrimidine with the ribose sugar, and ribose sugar is attached with a phosphate group at position 5 or at position 3. यानी कि यहाँ पे फिफ्थ पोजीशन पे भी एक फॉस्फेट ग्रुप अटैच हो सकता है और राइबोस शुगर की थर्ड पोजीशन पे भी एक फॉस्फेट ग्रुप अटैच हो सकता है. ये 1 है, दिस इज 2, दिस इज 3, दिस इज 4 एंड दिस इज फिफ्थ कार्बन. तो एस्टेरिफिकेशन के थ्रू हमारा जो फॉस्फेट ग्रुप है वो न्यूक्लियोसाइड के साथ एट फिफ्थ पोजीशन अटैच होना चाहिए. देन इट इज कॉल्ड एज अ न्यूक्लियोटाइड. This is the structure of ribonucleotide. And now syn and anti-conformation. Conformation means the change in the shape of any molecule when it is rotated around its axis. कि जब भी कोई molecule अपने axis के इर्द गिर्द rotate करेगा तो आप कहेंगे कि वो अपनी shape change कर रहा है, and it is present in different conformations. There are two conformations, syn and anti-conformation regarding nucleotides. Anti-conformation means के molecules की अरेंजमेंट ऑपोजिट साइड्स पे है, एंड सिन कन्फर्मेशन मीन्स मॉलिक्यूल्स की अरेंजमेंट जो है वो सेम साइड पे है. As polynucleotide molecules, they are large in structure, so there is no rotation around beta glycosidic bond. यानी कि यहाँ पे किसी भी किस्म की आपको रोटेशन नज़र नहीं आएगी क्योंकि हमारे पास नाइट्रोजनस बेसिस का और राइबोस शुगर का स्ट्रक्चर बड़ा है, जिसकी वजह से ये अराउंड द एक्सिस इजीली रोटेट नहीं कर सकती. तो नेचुरली आपको दो कन्फर्मेशंस नज़र आएंगी, एक एंटी कन्फर्मेशन नज़र आएगी और एक सिन कन्फर्मेशन नज़र आएगी. एंटी कन्फर्मेशन में हमारे पास दिस इज़ एंटी कन्फर्मेशन and this is anti-conformation, purine anti-conformation and pyrimidine anti-conformation. In anti-conformation, smaller molecules above the sugar ring, and in syn conformation, larger molecules are present in the syn position. Now this is syn conformation of pyrimidine, as you can see, and purine, and this is anti-conformation of purine, and this is anti-conformation of pyrimidine. Naturally, there is more anti-conformation of purines and pyrimidine rather than syn conformation. Now you have studied the basic definitions of nucleosides, nucleotides and the nitrogenous bases. This is the cumulative table of purine bases, ribonucleosides and ribonucleotide. This is the structure of purine and pyrimidines. This is the structure of adenine. When there is attachment of ribose sugar, it is called as adenosine. And when there is addition of a phosphate group, this is called as adenosine monophosphate. अब आप यहाँ पे देखें नेम भी बदल रहा है. That is single nitrogenous base, it is called as adenine. When there is an addition of ribose sugar, you will call it as adenosine. And when again there is a phosphate group, this is called as adenosine monophosphate, और आप इसको AMP से abbreviate करेंगे. This is guanine, addition of ribose sugar guanosine, and addition of phosphate group guanosine monophosphate. This is cytosine, addition of ribose sugar cytidine, and this is addition of phosphate group cytidine monophosphate. Then uracil, addition of ribose sugar uridine and uridine monophosphate. Then thiamine, thymidine, and thymidine monophosphate. Modification of polynucleotides can generate additional structure. Regarding nucleotides, you have studied the structure of purines and pyrimidines, and you have clearly see the structure of purines and pyrimidines, in which there is thiamine, cytosine, uracil, adenine, and guanine. If there is a changing of the structure of purines and pyrimidine, then we will call it as modification of these nucleotides. And this modification is present in nature. यानी कि आपकी प्यूरिन और पायरामिडीन जो हैं, जो कि बेसिकली हमारे डीएनए और आरएनए का पार्ट हैं, उनमें कहीं ना कहीं किसी ना किसी पोजीशन पे कोई डिफरेंट ग्रुप अटैच हो जाता है, तो आपकी प्यूरिन और पायरामिडीन मॉडिफाइड हो जाती है, और ये नेचर में प्रेजेंट हैं. मेनली दे आर द पार्ट ऑफ माइक्रो ऑर्गेनिज्म लाइक डीएनए ऑफ वायरसेस, आरएनए ऑफ वायरसेस, DNA RNA of bacteria, and DNA RNA of fungi. Examples included 5-methyl cytosine. 5-methyl cytosine is present in bacterial and human DNA. 5-hydroxymethyl cytosine, it is present in bacterial and viral nucleic acid. Mono and dimethylated adenine and guanine, they are present in mammalian messenger RNA, and it has role in oligonucleotide recognition and in regulating the half lives of RNA. जो RNA हमारी बॉडी में बनता है, उसकी फर्दर जो है मॉडिफिकेशन होती है. आप जब सेकेंड ईयर में जाएंगे तो उसकी मॉडिफिकेशन आप तब स्टडी कीजिएगा कि किस तरह से उसकी मॉडिफिकेशन होती है. तो ये जो स्ट्रक्चर है उसकी मॉडिफिकेशन के लिए मोनो एंड डाई एंड मिथाइलेटेड एडेनिन गुआनिन मैसेंजर आरएनए में वो काम करता है. देन फ्री हेट्रोसाइक्लिक बेसिक्स इंक्लूड हाइपोजेंथिन, जेंथिन एंड यूरिक एसिड. These are the intermediates of catabolism of adenine and guanine. जब adenine और guanine ह्यूमन बॉडी में ब्रेक डाउन होते हैं, तो वहाँ पे थोड़ी सी एडेनिन और गुआनिन की मॉडिफिकेशन होगी, और वो हाइपोजेंथिन, जेंथिन और यूरिक एसिड की प्रोडक्ट्स जो करती हैं वहाँ पे. एंड मिथाइलेटेड हेट्रोसाइकल्स ऑफ प्लांट्स, जिसमें कैफीन है जो कि कॉफ़ी में प्रेजेंट है, थियोफाइलिन है, ये भी मॉडिफाइड न्यूक्लियोटाइड बेस है जो कि टी में प्रेजेंट है, और थियोब्रोमीन है जो कि कोकोआ में प्रेजेंट है. दिस इज़ द स्ट्रक्चर ऑफ दीज मॉडिफाइड बेसिस. 5-methyl cytosine. अब आप देखें कि 5th पोजीशन पे साइटोसिन के मिथाइल ग्रुप की एडिशन हो रही है. देन 5-methyl हाइड्रोक्सीमिथाइल साइटोसिन के साइटोसिन की 5th पोजीशन पे देयर इज एन एडिशन ऑफ हाइड्रोक्सी मिथाइल ग्रुप, एंड दिस इज डाईमिथाइल अमाइनो एडेनिन के यहाँ पर 2 मिथाइल ग्रुप की एडिशन. 7-methyl गुआनिन के गुआनिन की 7th पोजीशन के ऊपर एक मिथाइल ग्रुप की एडिशन हो रही है, और ये हमारे पास नेचुरली अकरिंग प्यूरिन्स और पायरामिडीन्स हैं. बट रिमेंबर, दीज आर अनकॉमन. These are not the part of regular DNA or RNA. They are present in nature and they have performing different types of role. Most of them are present in microorganisms like viruses, DNA and bacteria I already mentioned. These are the regular structures of adenine, guanine, thymine and cytosine, and from there you can compare the modifications of these nitrogenous bases. This is the structure of hypoxanthine, xanthine and uric acid, and in previous slide I already mentioned these are the breakdown products of purine catabolic pathway. Characteristics of nucleotides. Nucleotides are polyfunctional acid. Now, you have studied only the structure of one nucleotide. They are present in poly form in DNA and RNA. In next lecture, we will discuss about that. And they act as acid, and they have pK value of about 1 and 6.2. In chemistry of protein, you have studied the pK values. What are pK? So you should be familiar with the pK values, and then they can easily donate and accept a proton at a physiologic pH. And there is another property of nucleotide, that they can absorb ultraviolet light. When they are studied in ultraviolet light, due to the double bond, conjugated double bond of purine and pyrimidines, they can absorb ultraviolet light at 260 nanometer. From this parameter, we can analyze, or we can study the structure of DNA and RNA. If it is present in um, an extract in labs, we can analyze the structure of DNA RNA by measuring its light absorption at 260 nanometer. Thank you for today. If you have any question regarding today's lecture, then please let me know in the comment section. And today's lecture was from the book of Harper Biochemistry and Lippincott Biochemistry, uh, 31st and 7th edition respectively.